Okay, well, welcome everybody to the Cellular Dynamics International webinar series. Today's topic, we'll be talking about the use of iPSCs, so induced pluripotent stem cell-derived cardiomyocytes, as a mechanism to study the genetics of left ventricular hypertrophy. Today's speaker will be Dr. Ulrich Patel. He is a professor at the Department of Pediatrics at the Medical College of Wisconsin, the University of Wisconsin. And with that, we'll just jump in. The webinar outline is shown here. Will commence as basically, I'll give a brief CDI overview and an overview of the iCell cardiomyocytes. Blake Anson, I'm the iCell cardiomyocyte product manager. Dr. Patel will then talk about using these cells in to study left ventricular hypertrophy. And then following that, we'll have a question and answer session. And actually, the question and answer session will take place offline. The webinar here will be posted on our website at Cellular Dynamics, and you can submit your questions through the question field there. So to start, we've got here an overview of cellular dynamics. We are the world's largest producer of human-induced pluripotent stem cells and the various cell types that can be derived from them. We currently employ about 107 people in Madison, Wisconsin. We've got three primary core competencies that are the foundation of the company. First is the creation and culture of the human iPS cells, the genetic engineering of those cells, and the manufacturing of the various cell types. So, for example, we're very well versed in the reprogramming of terminally differentiated cells to generate the iPS cells, and then we can genetically engineer those cells to express various biomarkers or use that as a way to increase our purity and our differentiation of the various cell types. And we currently offer 4 different cell types as products here. The cardiomyocytes, which are our first commercial product, the endothelial cells, which were launched in the Q3 of 2011, neurons, which are launched in Q4 of 2011, and our hepatocytes, which are under a pre-commercial evaluation. We also have a 5th product offering, and that is a services whereby we can reprogram from interesting genotypes or phenotypes that customers may have, and then generate iPS cells from those, do whatever type of genetic engineering may be required, and also differentiate those into any of the 4 cell types I just mentioned. But today we're going to primarily talk about the iCell cardiomyocytes product. These are human-derived. They're very pure, roughly high 90% purity. They come cryopreserved, ready to use. Whoops. We have inventory so that are available in virtually unlimited quantities. They provide a full product solution where we can have the cells, the media, and the protocols for your immediate use. And as you'll see in these next few slides, they really demonstrate normal human cardiac biology, and they show the expected responses. And we've developed them such that they can be used across a broad platform, a broad variety of platforms for discovery and preclinical development. All right, a little bit of basic characterization here. In this case, focusing on electrophysiology. We've got the ionic currents over here on the left-hand side. We've got the sodium and calcium currents necessary for depolarization, potassium currents necessary for repolarization, and then the funny current and IK1 that are required for rhythmicity, as well as setting that maximum diastolic potential. The net result of these currents is the generation of spontaneous action potentials, as seen up here in the upper right-hand quadrant. Each vial of cells contains a pan-population of cardiomyocytes that show atrial-like, nodal-like, or ventricular-like action potentials. In the lower right-hand quadrant shows that they've also have the appropriate GPCR pathways, where we have the beta-adrenergic, alpha-adrenergic, and muscarinic pathways. So we've got all the electrophysiological workings of functional native cardiomyocytes. We've also got a brief overview here looking at metabolism and contractility. The upper half of the graph illustrates that simply that these cells use their cardiomyocytes, that use their mitochondria, shown here under baseline conditions, and then if you modulate that by either uncoupling electron transport from oxidative phosphorylation or by shutting down the mitochondria, you see that oxygen consumption will either go up or down. So the mitochondria are active and can be modulated. The lower half of the graph of the slide here illustrates the contractility. That is that it's driven by calcium. On the left-hand side, you can see as we can measure calcium transients, and as those are blocked by nifedipine, we see contraction as measured by the ionoptic system. On the right-hand side, the contraction also decreases. So we've got a physiological metabolism and physiological contractility. As I mentioned earlier, we've really developed these cells to be used across a wide variety of platforms. These include any type of cell-based assays, numerous high-content imaging devices, and then with these platforms, you're able to look at the basic mechanisms of functionality, be it energetics, calcium handling, various electrophysiology, as well as then the contractility. So from a basic platform side, the cells are showing high utility. We're also able to do a little bit of disease modeling with these cells as well. And the topic of today's discussion will be hypertrophy. As we can see, hypertrophy is basically a small heart through a series of induction events is generated into a larger tissue. And there's many different outputs for measuring the hypertrophy. It can be protein synthesis, cell size, or the fetal gene expression. What we've done at CDI is generate a fairly basic model for looking at hypertrophy. And that is where we use endothelin-1 as an inducer, and we assay the output by looking for increases in the NPPA, NPPB mRNA, or the resultant protein levels. We can do that through qPCR, various microscopy, or flow cytometry methodologies. So we've got a nice, robust assay, but it's still very, very simple. What we're going to talk about today is Dr. Patel will talk about really taking these steps, or these cells, to the next step and looking at how this model can be used in true left ventricular hypertrophy. And so with that, I'll turn it over to Dr. Patel.
Well, Blake, great. Thank you very much for the nice introduction and the introduction of the cardiomyocytes and stuff. It's my great pleasure to give an overview of the research we're doing here in my lab using iPS-derived cardiomyocytes. Just to disclose this, first, my conflicts of interest. Our work is supported and funded by NIH, a number of grants there, as well as some support from the Wisconsin Genomics Initiative and from our institution. Let me give you a brief overview of what we're doing in my lab. We are obviously interested in identifying genes for complex diseases and mainly cardiovascular disease, so we're interested in hypertension, diabetes, and the complications of this. And when you look at this, where genetic studies stand today in humans is that we have over the last few years, accumulate a large amount of information from genome-wide association studies. We identified many, many loci there. And as we're moving from genome-wide association studies to genome sequencing, exome sequencing, whole genome sequencing, I think it's not very difficult to predict that we will just get a substantially large set of data. And but what this data really only gives us ultimately only statistical evidence that certain genes or certain variations, certain polymorphisms are associated with the disease. And while we have a large amount of data, we will have just more questions than after we've done these studies, because again, this really only provides statistical evidence to some degree, and it really lacks some of these functional annotations. So the questions what you can ask yourself is, after you did some of these studies, and you identified a gene, you can ask yourself, what does this gene really do? What's the function of this gene? And then even more importantly, as you might want to translate this in therapeutics, is, what can we do with this gene? Can we modify this? Can we influence the effect and function of this gene to potentially prevent the disease development? To some degree, and then there's a lot of active research going on right now, these studies, sequencing studies, obviously provide a lot of information which you can use diagnostically, because you really don't necessarily have to know the function of a genetic DNA variant. But it's really not very clear if you can also then use this for treatment, because again, I think to move this into the treatment realm, you really have to know what this gene does, understand the function, and study this in the context of the diseased tissue. So overall, that kind of leads us to the next step. What do we really want to do? What do we really need to do as a follow-up? And it's clearly we have to develop platforms to understand the disease mechanism. Ultimately, if we're looking at an individual patient with a disease, we want to understand each patient's disease characteristics to potentially develop new treatment options. And it really leads us to the theme of individualized medicine. And all this together, I think, means that we have to model disease in an appropriate way, which will allow us to screen for drugs or even replace and regenerate a certain tissue, which is to start doing disease mechanism. I think all those things really then guided us towards using iPS-derived tissue for the functional analysis and its follow-up for our genetic studies. So let me tell you a little bit about the disease, some of the diseases. One of the main research focus we had in the lab, or we have in the lab for many, many years now, and that is left ventricular hypertrophy. Left ventricular hypertrophy is really defined as an increase of the weight of the left ventricle. And it actually is reasonably frequent. Main causes for this is main underlying causes for left ventricular hypertrophy as a main complication. It's hypertension, high blood pressure, kidney disease, diabetes. Advanced age is a risk factor, as well as gender and race. In addition to that, there are a number of metabolic diseases which cause left ventricular hypertrophy, as well as some valvular diseases. So now, how do we really measure this? A lot of patients normally don't know if they have hypertrophy. And obviously you can't see this, but the way how it's measured is doing an heart ultrasound. So you see here some pictures on the right side. You see basically a slice through the heart. As you can see it in the ultrasound, you can measure the ventricular septum. You can measure the thickness of the ventricle. You can measure the size of the left ventricle, posterior wall, and from these measurements you can calculate the left ventricular mass or the weight of the heart. And at a certain point, when this exceeds certain normal values, then you speak of left ventricular hypertrophy. Now, why would we be really interested in this disease? And here's a summary of data from a large epidemiological study, or a large trial from the LIFE study. And they looked at individuals or participants with and without hypertrophy. So in solid, you have individuals with left ventricular hypertrophy. The lighter line is without normal LV mass, and you can see for a combined cardiovascular endpoint as well as for a very strong endpoint of cardiovascular death, as well as all-cause mortality in this cohort over 2 to 3 years of time, you see a significantly increased risk for cardiovascular death, all-cause mortality, as well as a composite cardiovascular endpoint in patients with left ventricular hypertrophy. So that really shows that we're talking about a clinical relevant disease entity. So now, what are the evidences that actually genes and genetic factors play a role? There's a number of studies, and over many, many years have accumulated quite a convincing set of data that the degree of left ventricular hypertrophy and the risk of hypertrophy is also determined by genetic and inherited factors. First of all, there's quite some variability there. If you take two individuals who both might have the same blood pressure level, one individual might develop hypertrophy, the other one doesn't. If you go in families and you look at familial clustering, that's one of the main ways how to determine and look at heritability and the role and identify the role of genetic factors. You clearly see a correlation in families that really directs the genetic component. There's also a very distinct subset of individuals who have normal blood pressure, but also have increased LV mass and have, that's what we call normotensive LVH. And in addition to that, from various animal studies, this also shows that there is a significant genetic component playing a role in the myocardial process, which leads to left ventricular hypertrophy. So from that point, it actually does make sense to pursue genetic studies. And so just a very brief introduction of the study cohort. What we had a long ongoing study in collaboration with the Hypergen cohort. This is part of NHLBI Family Blood Pressure Program. And the study design is a family-based study, so we have two siblings with hypertension, as you can see on the left. And we basically recruited all available additional family members here, and they were characterized in the echocardiograms. Overall, it's a biracial study, so we have African Americans and Caucasian participants, and we have about 1,200, 1,300 African Americans and slightly less Caucasians. Now, the approach to really identify genes has been the hallmark has been genome-wide association studies, and we performed genome-wide association study. In that full cohort, we did a family-based association and recently published our reports and findings. And we found a number of SNPs and variants which are associated with various phenotypes related to wall thickness as well as left ventricular mass, as described in this publication here. And as what I told you in the beginning, we're really now interested in trying to better understand what these genes and these SNPs, what these functionally do, and to do functional follow-up. So the next step then obviously is that we have to develop a disease model which recapitulates hypertrophy, and we obviously want to do this in human cells. It should recapitulate the disease, and that should potentially then allow us to test for treatment and to develop novel treatment options. And so this is then when we started to use the cardiomyocytes. So the first step here is showing you data on how we induce hypertrophy in these cardiomyocytes, and as Blake mentioned this, there are a number of ways how you can do this. So we did our initial experiments looking at a fixed dose, but we stimulated those cells either with isoproterenol, endothelin-1, or angiotensin-2, as you see in the figure A. Compared to controls, all these stimulants clearly show an increase in the cell size of these cells. The way how we measure this initially is just to characterize it better, just look at the cell size. Increase in cell size is also the correlate what we see in individuals and in the normal human heart. So if you look at on the right side, we also did some immunohistochemistry where we stained for CFOS. It's a key characteristic gene, protein, which has increased in hypertrophic cardiomyocytes. And again, you can see the stimulation with isoproterenol, endothelin-1, or angiotensin-2 shows an increase in the CFOS intensity. So overall, these cells do develop hypertrophy, and they have characteristics of an hypertrophic state. And I also want to mention that isoproterenol, endothelin, as well as angiotensin are factors hormones which have been shown in a number of studies to be associated, linked, or even elevated in patients with this disease. So those are actually quite physiological stimulants. So the next step then what we did is we also performed an expression array, an expression analysis to look at the global gene expression of this. And in the upper right corner, you can see the differential expression in the different stimulation protocols for isoproterenol, endothelin-1, as well as angiotensin. And you can see here that there is some overlap between those, but there are also some unique genes which are uniquely differentially expressed in a different experiment. But we also have a subset of genes which are actually a few of them, about 5, which are differentially expressed independent of the stimulation of these different receptors. And to some degree, that is expected because each of these drugs stimulate obviously different disease pathways. So overall, now our design is, we do a family-based association study. We're starting to actually recruit additional family members and develop patient-specific iPSC-derived cardiomyocytes, from them. But I also want to show you then some results really addressing the question, these are, iPSC-derived cardiomyocytes. How do they really resemble what we observe in a patient or in a human being? And obviously, a cell culture is a cell culture, and a whole organ is a whole organ. So we do have to recognize that there are differences. But we try to address this concern or this topic, and again, looking at expression differences. And here, to show you the results of this, one of the ways how we look at this is actually where we performed a principal component analysis, and we're looking at a subset of genes which are differentially expressed, and we have two main data sets. So we have our main data set on the right side here, in blue and in kind of like the darker yellow, which are the cells, which are the isoproterenol stimulated cardiomyocytes. In purple, the stimulated one, and in blue, the control cardiomyocytes. Now, we were fortunate enough that there's a public data set out there, and this is a expression analysis performed on cardiomyocytes which have been obtained through a cardiac biopsy, and that's done, actually, that was done in patients in green in control individuals, as well as in red and in light pink here in individuals with hypertrophy, LVH, in light pink with normal ejection fraction, normal heart function, and in the red with LVH, but a reduced ejection fraction. Those are patients with heart failure. And so we basically clustered this and said, can we separate those samples? Can we separate that, and are there components, subset of genes, which differentiate that? And principal component 1, the factor which separates this most, is obviously a factor which separates the iPSC-derived Cardiomyocytes, the normal ones as well as the stimulated ones. It separates those from the patient samples. And that's as expected, because as I said before, a cell culture is a cell culture, and organ is an organ, so we do expect that there are certainly differences between those. But the more interesting thing is that the next strongest principal component really separates out the normal state and the hypertrophic state. So towards the negative one, those are principal component clustering with the hypertrophic response and expression pattern. And on the upper part of the diagram, you can see here in green and blue, you see the normal cells. So you see here also the normal patient cells grouped together in this component, together with a normal iPS cardiomyocyte. Now, there are a number of different analyses we can do, and it basically more or less shows the same, that these cells actually do, in fact, when we compare this to data from human hearts, that the expression really resembles it and really does, in fact, reflect the disease phenotype. So the next step here, and this is a little bit of a busy slide, let me explain that to you. We're looking, one of the ideas is that we can actually now combine the evidence from our genome-wide association study, from the genetic evidence, and we want to combine this also with the evidence what we have from our expression arrays. And common sense would say that a gene which has a positive association from a genome-wide association study, obviously that plays, has a high chance of playing a role in the disease process. And we would also say this for a gene which is differentially expressed. So if a gene which is differentially expressed, quite likely that it contributes or plays a role in the disease process. And so one of the questions you can ask yourself is, well, could you combine the evidence, both from genome-wide association as well as from expression, and really come up with an overall likelihood? So can we identify then even a subset of genes where we have association evidence and also evidence for differential expression, because these genes it would make sense that they are even more likely to play a role in the disease process. And so one of the technologies or methods how you can do this is called gene set enrichment analysis, and this is what we did here. On the plot here, just to explain this to you, if you look at the green line, the black lines underneath the green graph here, each line basically represents a particular gene. And so what this analysis does, you basically add genes with the high evidence of differential expression, high evidence for association, and you include this in the model. And the green line really kind of represents a overall likelihood of your model. And you start adding genes to this model until you reach a certain peak. And then at some point in time, you add more genes, and then the overall model becomes less likely, and then you basically, the green line goes downhill again. So everything to the left of the peak of your model are basically then genes where there's evidence that they contribute to the model and they have an effect. Then to the right, these are genes which do not play a role anymore. So we have a number of genes that are really kind of clustering here where we can identify them as highly likely candidates and that they really do play a role in the disease process. And obviously, with this functional and association evidence, those will be then prime candidates for further functional screening down the road. Now, then you could ask yourself is, how can we screen for the effect of this? And obviously, there are a number of different ways, but we just want to discuss here two different ways. Obviously, you can do a drug screening where you can either take known drugs. That's obviously a very quick approach. If these are drugs which are in the market, you can use those and test if any of these drugs have additional effects on your disease of interest. Or you could, for example, do a compound screening to identify new compounds and have a new mechanism. Obviously, if you do take known drugs or compounds, you're probably much quicker towards the treatment, develop a new drug, and it's obviously what we would like to do. In many ways, though, the negative thing for this one is that the mechanism, how these compounds, particularly in compounds, how these compounds really work or affect your gene and phenotype is less clear and needs a lot of workup. The other way, and this is what we pursued here, and I'll show you some data on this, is using SI small interfering RNA. There are now libraries out there, basically validated siRNAs for most of the genes in humans and as well as animal models, and it's a very targeted approach to silence gene or a gene which is overexpressed. I have to mention this, obviously. On the other side, again, if you're thinking about therapeutics, and although there is some momentum to use siRNA as a therapeutic or as a drug, the way to really come to a treatment obviously takes a little bit longer. But I think we pursued siRNA because we had a number of genes which really made sense, and we wanted to very specifically target those genes to see if these genes have an effect. And so just to very briefly review with you, siRNA basically are small strands of RNA that get transfected in the cells. They bind to RNA, and with their binding, basically the messenger RNA gets degraded, and in the cell it knocks down the microRNA and stops the production of a certain gene product. So this is the mechanism of this. So we developed a protocol for a siRNA transfection in these cardiomyocytes, and basically transfected these cells and targeted one of the genes which we identified in gene Set Enrichment Analysis. And so I'll show you the cells here, again looking at cell surface area for a number of different experiments here on this little busy slide. In gray, this is the cell surface area of the unstimulated cells. In red, you see the cell surface area of the isoproternal stimulated cells. And we did a number of controls to again control transfections to control for the reagents which are used in the transfection, in the siRNA transfection, just to make sure that those do not induce hypertrophy, as this is a certain stressor to some degree. And you can see here there's really no difference in the cell surface area in any of the four gray groups here. Now, if you look at the isoproternal in the first on the left, the second and third, again, these are the different controls. We see a significant increase in the cell surface area in these cells. And the one to the very right, this is the one with a targeted siRNA for one of our candidate genes here. And if you do a comparison where we take the one to the very right and compare this with all the gray ones, we do not see a significant difference. So basically, the cell surface area for the stimulated cells, but knocked down with this siRNA for one of the targeted genes that basically resemble the unstimulated cells. So to some degree, we can say that we block the development of hypertrophy, isopaternal-induced hypertrophy, by blocking this particular gene. Now, this is just one of those genes. We obviously screened a number of genes, and I only show you here the positive one. I could also tell you that we had knocked down a number of other genes and targets, and we didn't see that response. So, I'll save that, for time, but, not every gene we knocked down actually worked. So, overall, the other thing, what we're studying with this, and I just want to very briefly at the end mention this, we're also looking at MicroRNAs and their role in hypertrophy. So we're pursuing this, and MicroRNAs are really regulatory RNAs who regulate the expression of a number of RNAs down the road. They obviously have been described as key players in cardiac development. There are very specific patterns of microRNAs which are related to certain disease processes, not only cardiovascular, but a lot of other diseases. And there's really an increasing interest in microRNAs as drug targets and as well as for diagnostic markers, for example, even in heart failure and hypertrophy. So overall, what we did is there are two basic ways how to measure microRNAs right now. There has been, for some time, there has been a race specifically for microRNAs to look at differential expression. They mainly focus on known RNAs with the development of novel sequencing technologies. Obviously, sequencing is likely to be the technique of choice, and so we basically pursued sequencing also with the option of potentially then identifying novel microRNAs. And here, just very briefly, review with you some more preliminary results. We sequenced a normal and a hypertrophic sample, and we did find a number of differentially expressed or represented MicroRNAs, in red, you see here in the right upper figure, you see in the red significant ones, and we see a distribution of a number of different, potentially also borderline significant microRNAs here in a list of those. Some of them have been identified, and we're actually pursuing this right now for to do some functional validation. So overall, in conclusion, I hope I could show you that these iPS-derived cardiomyocytes actually provide a platform for the functional genetic analysis and genomic analysis, particularly if you're interested in really following up human genetic studies. I think they also offer, these cells offer tremendous opportunity to study patient-specific unique disease if you have very patient-specific cell lines. And obviously, I think the strength, what we see is really combining both genetic and functional data to really understand disease mechanism and then opening this up to development of new treatment options. And this was really the work, what I present here, really the work for a large number of people and groups here with whom I've been working together for a long time. And I really want to thank, on the left side, the people in my lab, and in particular, Andrea Metter, who developed and worked a lot on the iPS cardiomyocyte cell culture, as well as Rachel Kramer and Amy Turner, who worked on the sequencing and DNA analysis, as well as Alex Stott and Praful Agarwal on the bioinformatics and